Hello, in this tutorial I will go through how to use LAM to analyze samples and explain the different functionalities. For this video, it is expected that all sample data has been organized as explained in the video concerning input, and that all samples have vectors as discussed in the previous tutorial. Most aspects of analysis can be controlled through the graphical user interface, which should be enough for most use cases. The default setting values of GUI are defined in settings.py found within the src folder of LAM. Editing values in the settings.py will consequently change the GUI's default values. The settings file also contains explanations for the different variables, but also has some not often used hidden variables that cannot be found in the GUI. LAM also accepts some setting changes from the console, allowing creation of batch files for consecutive analysis without additional user input. For example, when whole midget samples have been split into smaller subsets. This will be detailed more in the next tutorial regarding split and combine. In the upper frame of the GUI are the primary settings that enable all functionalities of LAM. The process option controls the creation of sample vectors. The count option projects datasets onto the vectors and then counts occurrences. The count option must always be performed for the data. The count option also anchors the samples when MP data is available and use MP is checked. The MP label variable defines what data folder the anchoring points are found in. Users should always input a name to an existing data channel in the vector channel setting, as it is used for several operations such as width determination and border detection. The bin number defines how many bins each sample is divided into. Having fewer bins drops resolution, while having too many makes comparisons more difficult as binned variable values near 0. The default bin number is 62, which we have found a good approximation for whole midget samples. In the other window, you can control what data is collected during the count functionality and how it is found. The first field is the column name of the data, the second field is the name of the data file with the variable column, and the third one is the unit of the variable. The unit field accepts Unicode values when placed between $signs. The data columns and locations must be given here even when in the position.csvs. In the lower part of the window, you can order LAM to output the data with replaced identifiers. The header row setting should also be changed to point to the correct index row of column labels within data files for correct reading. LAM expects all input data to have the same column label row. The count setting also enables the width approximation of samples using the dataset defined in the vector channel setting. Variables related to border detection are controlled through settings.py. The quality of output cannot be guaranteed if the default border detection variables are changed, but better variable combinations and scoring may exist depending on desired outcome. The distance setting enables the clustering and feature-to-feature distance calculations, which are controlled by the bottom frame. Multiple channels can be given if separated with comma. In the feature-to-feature distances, another channel can be given as a target dataset for nearest objects, enabling calculation of distances, for example, from each delta positive cell to their nearest Prospero positive cell. In the clustering algorithm, objects are marked as neighbors when within the distance given in maxDist option. These object neighbors are later merged based on shared IDs. The min and max cell number variables limit the number of cells considered as a cluster. The dataset for the distance calculations can also be filtered based on whether they have smaller or greater variable values than what given in the filter value setting. The plot setting controls plotting of data produced by count, distance, and stats options. You can select all wanted plots from the plotting frame on the right. Some relevant settings are contained within the plots window. The drop outliers option only affects the plots. The fields in the versus plots section control what data is plotted in the bivariate plots, namely the channel versus add and add versus add plots. In the lower frame are the controls on how significances are marked on statistical plots. The stats option handles statistical testing of sample groups for all variables. Plotting of stats must be done simultaneously to the calculations and cannot be done on a separate run. In the stats window, we can set the control group, determine the significance limit, and control whether the statistics are calculated in a sliding window or bin-to-bin. The total statistics checkbox enables comparisons of pooled bin values of each sample group, and the group versus group checkbox enables group-wise comparisons along the length of the vectors. Once the settings are in place, the analysis can be started from the Run button. Depending on the circumstances, LAM might ask for user input during the analysis. The dialog can, however, be suppressed with the Force Dialog option that is found in settings.py. All LAM output is saved in the analysis data directory that is created in the analysis directory. In the data files folder, you can find most of the produced data, for example, files with the prefix all that contain the object counts at each bin, and files with prefix norm that also contain object counts but placed in the anchored data matrix. All calculated statistics are placed in the statistics folder named after their statistic type and used sample groups. In the plots folder, LAM places all generated plots with the exception of vector-related plots. The various plots are either placed in the root of the folder or in aptly named plot folders. Within the samples folder, LAM stores collected data of the samples in their own folders. Additionally, the vector-related plots are placed here. Within the sample folders are channel-specific data files that contain new columns depending on performed LAM functionalities. Better descriptions on the output files can be found in the user manual.